Hello again. Welcome back. I hope you had a good and connecting lunch. So this is our first session for presentations by next generation scientists. It's a rather novel format for Lindau. I think it's the second year. We tried it out last year. We are expanding it now. Previously we tried with poster presentations, but we think this concept is perhaps better. So very welcome, those of you who are going to present and those of you who are going to listen. My name is Claes Kärre. I'm an immunologist from Stockholm, Sweden, and I will moderate the session. And we have 9 presentations in 90 minutes. So with 1 minute transition time, that means 9 minutes for every presentation, 6 for the presenter and 3 to ask questions. If you want to ask questions, please line up either there or there, to the left or the right at the microphones. And I hope we'll have time for some questions after every talk. So our first, and I will be very brief in presentations in order to give as much time as possible to the speakers. So our first speaker is Hersh Bhargava, currently based in San Francisco. Welcome up on stage. The title is Engineering Principles of T-cell Proliferation Control, Computational Design of Circuits that Counteract Tumor Suppressants. Please.
All right. Thank you very much. It's a pleasure to be here. Thank you all for coming. I'm going to talk today and keep it simple and hopefully show a few movies as well, but discussing work from our group in engineering T-cells, hopefully to be better at treating cancer, but moreover to be more rational in our approach to engineering of these cell therapies. So the natural functions of the immune system are really the basis of human health, period. Immune cells have sophisticated functionalities involving the ability to regulate proliferation, killing of target cells, even your own bodily cells with single cell precision, the ability to migrate throughout the body in a targeted fashion, and also the ability to deliver payloads. And through the orchestration of these simple modules, immune cells in a healthy person affect the protection of people, hopefully like us, from disease as well as infectious disease and even cancer. But as we know, this system will not protect us forever. Eventually we will develop disease and all eventually die. So the question of synthetic immunology and cell engineering is, can we augment the immune system and take this natural scaffold and add functionality to this baseline such that we can achieve therapies for specific diseases or bolster immunity in general. So the paradigmatic example of this immune cell engineering is CAR T-cell therapy, where now it is a clinical paradigm to isolate T-cells, effector T-cells and CD8, CD4 T-cells from patients' blood, genetically modify them to express a receptor which retargets their natural activation and killing program against a arbitrary antigen, and then reinfuse the resulting recombinant CAR T cells into patients. This strategy has been highly effective in the treatment of certain blood cancers, and there are now 6 CAR T cell therapies approved, all for treatment of blood cancer in the United States. But stepping back from the specifics of extant cell therapies, there's a broader question, which is now we have the ability to introduce synthetic user-defined genetic payloads into immune cells and then put those cells into people. So the question becomes, as Francis Arnold said in the first talk, we now have this huge space, the universe, and maybe the cure for cancer does live somewhere in that design space. So what do we engineer T cells with, and eventually other cells as well? And there's this huge repertoire of all natural and de novo designed and directedly evolved proteins which we can use to build circuits in these cells, and in principle many of those can be actually delivered. So I will briefly touch on one specific case study in this space where we tried to augment CAR T cells, which ordinarily show limited efficacy in solid tumors, and in particular in solid tumors which are immune excluded. That is, you don't see much accumulation of immune cells in those tumors because those tumors have evolved mechanisms to suppress the natural immune system. So here we can show that if we use just regular CAR T cells, there's no response. These mice, this is a model of pancreatic cancer, these mice will die of cancer. And here you can see from CD3 staining, T cells don't even make it into these tumors. There's very minimal accumulation of engineered or natural T cells in these mice. We'd call this an immune desert or immune excluded phenotype. So we asked here, thinking back to synthetic biology and this space of possible ideas, could cytokines be one of the levers which we can use to push immune responses to be stronger in these tumors? And so with this fundamental idea, which is not a new idea, people have been injecting patients with IL-2 since the 80s, but we asked, can we potentially design a space of strategies to deliver cytokines to our cell therapies in different ways? And perhaps these can augment the functionality of T cells, in this case, delivering IL-2, which is a growth factor for T cells, in different paradigms and with different circuitry, ranging from the simple idea of injecting it directly into the circulation, all the way to building synthetic circuits with synthetic receptors. In this case, in the bottom, you can see we use these synthetic Notch receptors to recognize tumor antigen and induce a circuit that produces, in this case, IL-2. And so we used a combination of both mathematical modeling of these different circuit paradigms, as well as, of course, in vitro and in vivo testing of these different architectures to evaluate which circuit architectures would be predicted to be efficacious and also which ones would show efficacy in vitro and in vivo. I'll summarize the results for you and not go through the details of each and every circuit that we tested, but what we found was, interestingly, a few design principles which seem to be fundamental, and we found that this particular circuit, which is an inducible IL-2 producing circuit, robustly improved the ability of T cells to colonize immunologically cold solid tumors. Here you can see that this dramatically improved survival in the same pancreatic cancer model I showed you before, and by staining these cells, or rather staining the tumors, we could see significantly improved accumulation of cells in those tumors. Just to touch on some of the specific principles that we learned here, inducibility appears to be a key feature. If you constitutively produce IL-2 or saturate T cells with these cytokines, they adapt. Similarly, the orthogonality of this circuit, it's not suppressed by T cell exhaustion or suppression of T cell signaling, so that appeared to be important as well. So I'll end with a bigger idea, which is, many disciplines have undergone a revolution to rational design and a transition. Now we don't build bridges by guesswork and wonder whether it's going to hold the weight. We build it in silico and then build it in the real world and have predictive capability. So hopefully, maybe through the efforts of some scientists here, we can achieve something similar in the world of synthetic biology and cell engineering. So thank you very much for your attention.
Thank you very much. Very nice. Questions? If somebody wants to step up, I can ask the first question. You mentioned in the abstracts proliferation circuits, and
Yes.
IL-2, I guess, is a proliferation circuit, but that shouldn't necessarily make the cells go into the tumor, migrate to the tumor, but
Yes.
once they are there, they would, of course, expand in the tumor. Is that the idea
Yeah,
here?
so that's a great question and something that we're actively looking at. Is this an effect where a few cells get into the tumor and expand, or is there what we would call a pioneer effect, or it's in some other way advancing the ability of these cells to infiltrate the tumor? So we're doing sequencing of basically TCR diversity as a proxy for diversity of the progenitors within the tumor. So hopefully we'll have an answer definitively in a short time.
Question here. Thank you.
Thank you so much. I was wondering, how do you do can you comment on the modeling, how you do that? Because in combinatorics, we always have the problem that the search space is just so big. So how do you narrow this down?
Yeah, so that's been the fundamental challenge of applying quantitative models to synthetic and systems biology for years. I mean, parameter identification is difficult. There's often degeneracy in the parameters. Validation of these models is difficult. So I obviously didn't talk about it specifically here, but we employ an approach that basically involves sampling of parameter spaces and design spaces in the neighborhood of the experimentally measured values, and then making conclusions not about specific points in a parameter space or design space, but about directions sections and neighborhoods in that space, which is our effort to be more robust to the specific behaviors of individual points in that large space, as you point out.
Great, thank you.
Time for one more question. So you speak about testing the different circuits against the variable cold tumor environment. Can you explain that? How do you vary the tumor environment?
So one of the things in silico that we can do that we can't easily do in actual systems is, everything can be a continuous x-axis. So we have in some of these mathematical models the ability to model different mechanisms of suppression. For example, T-cell exhaustion, the rate of cell ingress into the tumor. So in silico is one thing. In the experimental side, we can also get at least points in the space of how cold these tumors are. For example, there are different tumor models we can use, which is changing multiple things at once, but we know that some tumors will recruit suppressive immune cells such as Tregs or M2 macrophages, and so those can provide a model where we have a more suppressed tumor versus one where there's more maybe accumulation of bystander T cells, which are not effective in clearing the tumor, but at least there are CD4, CD8 conventional T cells present.
Thank you. 904, very good. That's a really good role model there. Thank you, Hersh.
Thank you.
I invite the next speaker up on stage, Stefania Boneva.